Hello and welcome to the exhibitor workshop from RainDance Technologies. I'm Rhim Banerjee, President and CEO at RainDance. We appreciate the opportunity to have you here. I see several familiar faces in the audience. Great to see you all. Today, a very special day. We've announced recently the launch of our Thunderbolts Cancer Panel, which runs on our Raindrop digital PCR system. I'm pleased to be able to speak to you about this. We also have an illustrious group of key opinion leaders who will be presenting today on data that they've generated on both Raindrop digital PCR as well as on the Thunderbolts Cancer Panel. So just to provide you a few slides of quick background, I think for those of you who don't know us, we are a core technology platform company based on digital droplet technology. We can take biological samples and convert them into single molecule droplets between 1 million and 10 million droplets a sample that runs on our systems in a highly automated, simplified manner with ultra-sensitive detection and true single molecule sensitivity. We've taken an open chemistry and open assay approach in that we run with any of the existing next-gen sequencing systems, as well as any hydrolysis probe-based PCR chemistry system. We provide flexible content and custom content capabilities to many of our research customers. We are automated, multiplexed, and ultimately low cost. We have a broad range of applications across our portfolio, notably commercially in digital PCR as well as next-gen sequencing content enrichment. To provide some context in what we as a company are trying to do, if you think of the world as I did in cancer sequencing along two axes, the number of targets versus the sensitivity achievable per target, the world of NGS has really expanded along the x-axis, increasingly more number of targets, but with a decrement in sensitivity as we get out towards whole exomes and whole genomes. PCR has gone the other direction, vertically, greater sensitivity, but limited by scalability of targets. Digital PCR and offerings from other companies have certainly expanded some of the capabilities of the qPCR market. However, our focus at RainDance is really about expanding the entire frontier. And so these are real numbers, things we're doing with our Raindrop digital PCR system, as well as our Thunderstorm next-gen sequencing content enrichment system, opening up new paradigms such as fluid biopsy, being able to detect low-frequency alleles and low-frequency mutations from tissue, FFPE, circulating tumor DNA, for example, circulating cell-free markers in general, as well as new forms of fluid biopsy. We're doing this at a much lower cost than has previously been possible, and importantly, creating access to genetic information that's previously been inaccessible. As a company, we have three major product areas. The first, our Raindrop digital PCR system. Just to give you a quick schematic, think of this as from sample extraction, our Raindrop source instrument generates droplets, and that's where the PCR reactions get put in with your genomic templates and ultimately are read out through the Raindrop sense, which is true single molecule detection. The Raindrop for digital PCR is the most sensitive single molecule detection technology for nucleic acids, period, full stop. Thunderbolts, which is the new cancer panel we've just launched, actually does content enrichment on the Raindrop source instrument to dropletize both your genomic DNA and primers into these microdroplets. However, the readout is today on a MiSeq at 2x250 paired end reads, and eventually with the HiSeq reaching the longer read lengths, we anticipate moving to the HiSeq as well. And meanwhile, the Thunderstorm system, which runs with next-gen sequencing content enrichment, works with much larger panels and is a much more high-throughput automated solution for larger laboratories, which then can be read out on any of the commercially available next-gen sequencers. As a company, I wanted to share with you some of the inherent advantages of our Thunderbolts product that runs on the Raindrop source and then is read out on an Illumina sequencer. First is it's a broad array of content, so 230 amplicons covering 50 genes. These are some of the most important and known cancer hotspots, tumor suppressor genes, as well as drug resistance markers. We feature superior coverage, so 100% coverage at a read depth of approximately 2500x. And as you'll see, we get 100% coverage even at 100x or 100 hits on the same target for sequencing. That's something that no other company has been able to achieve or claim to achieve out there. We start with low starting sample amounts, so as low as 10 nanograms of DNA with no preamplification. This is really important for precious and scarce samples, particularly FFPE or circulating cell-free markers. We've worked and been successful with a whole range of tissue types, FFPE, biopsy tissue, and also with plasma. And you'll see some of that data from our key opinion leaders today. We work with the predominant Illumina next-gen sequencing systems, but importantly, we have the sequencing adapters and indexers integrated directly into our primers, such that there is no library prep involved in the workflow. Importantly, when you roll this up in real-world terms, it's less than 3 hours hands-on time with total results in under 2 days, with the MiSeq's 37-hour run being the primary determinant of that long time frame. It is ultrasensitive. We are seeing minor allele frequencies south of 5%, and that's something that's relatively unique to the product. And importantly, we wanted to be able to democratize this market. So you can get down to between $100 and $150 a sample, including cost of sequencing, including the all-in workflow, depending on whether you're using a MiSeq or a HiSeq, with no upfront setup costs. And importantly, all of this runs on our low-cost raindrop source benchtop instrument, which is priced at $50,000. Of course, your sequencer, your MiSeq or other sequencer is on the back end of that. We cover some of the most important hotspots in the cancer amongst oncogenes, and I'll leave this for some of our key opinion leaders today to cover. And importantly, when we first launched our first access program in January, you can see here very strong data across the board for working with two collaborators, Tony Godfrey at Boston University and Pierre-Laurent Puig at the University of Paris. You can see for a tumor quad set, as well as FFPE and fresh biopsy, all the way down to 10 nanograms, we saw excellent numbers of excellent coverage. We also saw out at 100 hits per target, exquisite coverage out to 98, 99% across the board, and also very strong uniformity, as you can see here. And this was a beta panel that we had launched at the time. The data you'll see from the key opinion leaders today reflects the further improvements on this beta panel. And today we have, over the course of today, 4 presenters, 3 of whom will be presenting at this workshop. So Dr. Andy Brooks from RUCDR at Rutgers, Dr. Fraser Simons from MD Anderson, as well as Dr. Andy Rhim from University of Michigan. And I'll do their introductions here in just a moment. But one more thing, which is while they're going to be presenting data on the beta panel that we had provided them about a month to 5 weeks ago, we've also not stood still. So we've also continued to improve on that panel. So here's the latest data for the commercial launch panel. You won't see this reflected in the data from the KOLs today, but this is the latest data from our commercial launch panel that we're making available as of this conference, that you can speak to any of the RainDance representatives here in the back of the room if you'd like to get more additional information on ordering. But you can see here for fresh frozen and for FFPE samples, all the way down to 10 nanograms, very nice overall coverage. And then as you look at the overall sequencing coverage, even out at 100x hits per target, significant coverage here at 100%. You can also see the extremely high uniformity as we've optimized this panel. And you can see even out at 500 hits in the fresh frozen, 100% of all the samples are covered at 500x. And in the FFPE case, I believe there was 1, maybe 2 amplicons that had started to drop out a bit. But you can still see exquisite coverage with very low FFPE samples. So with that, let me turn over to some of our key opinion leaders who will be presenting today. And first, I'd like to welcome Dr. Andy Brooks, Chief Operating Officer from Rutgers Cell and DNA Depository. Andy is very well known, I think, in the industry, and today will be speaking to us about his research in colorectal cancer. Thank you, Andy.
Great. Thank you, Rupam. Good afternoon, everybody. What I'd like to present over the next 15 minutes or so is our experience with some of the technology that Rupam had just described. And our implementation for technology like this is a little bit different than most laboratories, given that we function not only as a large sharing biorepository, but also an analytical services group that generates not only high-quality biomaterials, but also data, which is distributed to a large number of individuals. So our responsibility for getting it right, so to speak, is a little bit larger than from an individual lab level. So just by a brief way of introduction, the RUCDR is currently the federal repository for 5 NIH institutes. We enable the sharing of DNA, RNA, cell lines, tissue, and data, not only for NIH institutes, but for a number of research advocacy groups and private sector clients. We have a large staff that has to be trained in these technologies, so technology implementation is a challenge for us, depending on what we're trying to do, and we have very large numbers of samples. More recently, we have built a CLIA lab within the repository for starting to expand some of our applications so that the data could be more broadly used. And the perspective that we take when it comes to every sample being treated as a limiting or precious sample is that there's a difference between just storing your samples and biobanking a sample. So for us, every sample that we take has to be considered really kind of a renewable resource, meaning that many labs have access to these samples to do their own kinds of analyses. And even if we're going to provide data on that sample, people still want those primary samples to validate and generate their own information. So the difference between a stored and a biobank sample is that a bank sample is really proactively acquired for future testing and analysis. And our partner in the area for molecular oncology is the Cancer Institute of New Jersey, and their personalized medicine initiative is really to collect every sample that comes in the door, generate a set of baseline data that can then help research as well as some of their clinical goals down the road. A bank sample is often sent to multiple different recipients. So again, we talk about having very limiting amount of samples. So the technologies that we focus on really require that minimal sample be used so we can preserve those collections. And a bank sample should ultimately never be depleted. Within our program, there are 5 major functions, with the last being analysis. But sample acquisition, the processing of that sample, whether it be FFPE, fresh frozen tissue, whole blood, what have you, then the storage and distribution of that sample is critical for everything that we apply our technologies to. So a lot of us that just look at that end stage analysis really wonder about what that sample went through to get to where we are, and then how do you effectively assess the quality of your data based on that sample. So tighter, more reproducible technologies are really critical for success. Some of the essentials that we look for, which lead into the technologies that we run, are around these functional essentials that we apply for maximizing biological resources. We have to maximally use our primary sample because there are undefined applications. If 7, 8 years ago we were talking about next generation sequencing, we wouldn't really know the quality or the amount of sample that we would need. And even in this short period of time, those requirements have dropped. And just when you thought they're about leveling off, new technologies are developed that allow us to get even further with even less amount of sample. Efficient processing, maximizing our extraction technologies, defining appropriate storage formats so we don't squander precious samples, looking at creating renewable resources, creating cell lines, iPSC lines from cryopreserved lymphocytes and other things that we could look at for analysis down the road, as well as nucleic acid amplification, and then ultimately the appropriate distribution guidelines. So when you request a sample from a biorepository or somebody requested from you, make no mistake, they're making their own little biorepository with those samples. And we only have enough sample to go around. So when we started working with RainDance, it was actually around digital PCR for an application where we were looking to dig deeper in assessing the quality of our samples. And by this I mean, when I show you our DNA workflow in a second for analysis, we were really looking at quality control based on what the available detection technologies could tell us about our sample. And that really was limited to high throughput genotyping for a large number of these samples and these very large studies. Therefore, our quality control was looking at the level of genotyping. Now that we distribute these samples for much higher resolution analyses, it's our responsibility, and it will be lots of other biobanks and those that hold those samples for all your analyses to be able to look deeper. Well, one way we were able to achieve this is by looking at digital PCR. And we looked at many different technologies that could do this, and the fact of the matter is, as Rhim had alluded to, in order for us to really say something quantitative, not just qualitative about our samples, is that we needed to get the single molecule detection. So again, this was a slide that he had shown very quickly, but I just wanted to underscore that this kind of droplet technology when it comes to digital PCR allows us to qualify every sample for a key set of markers before they're distributed for deeper analysis. And then more recently, we looked at the Thunderbolt's target enrichment panel, so for the cancer panel. And what this allows us to do is, again, to use very small amounts of sample and using this droplet-based technology, actually get, again, a single molecule resolution, but this time not for PCR, but at the level of next-gen sequencing. And why is this important? It's important because we can actually engineer this kind of technology into our workflow. So it becomes a standard part of the collection, extraction, analysis, and storage part of our workflow, which makes this data available, which really short-circuits the time of being able to select the samples that you now want to do whole exome, whole genome sequencing on, and really maximizes the use of each of those samples. So these are the genes that are on the panel. They're comprised of about 230 amplicons. We had looked at this panel for a variety of different sample types. And one thing to point out is that we actually looked at this, and when we evaluated it, compared to the AmpliSeq panel, the Cancer Hotspot panel version 2, which we've been using for a long time, and wanted to see comparatively what those results look like. The targets selected here, I think were very well thought out. Again, for us, it's about continuity for samples. So we have a lot of data, a lot of legacy data, and we need to know that we can compare that data for new samples on platforms like this to what exists, especially because we keep this centralized database that we're responsible for all of the users for these samples. The nice thing about this approach for this technology for us is that we actually can use the same technology to validate our next-gen sequencing target hits. So it's the same technology. The source instrument generates the droplets. In this case, the Sense for digital PCR allows us to do the counting based on our qPCR measurements. It's just as simple as setting up Thunderbolts for generating your library, but here we actually get detection based on our qPCR outputs, and I'm going to show you the same technology was actually used to validate the hits in this evaluation. When we start implementing technology, there are a lot of considerations that we take, again, because of the scale and the size of what we're responsible for doing. So we have to look at specificity. So content selection and accuracy is key. We look at the sensitivity of our technology that we're evaluating, how much starting material do we need, what levels of detection can we achieve, how applicable is it to our programmatic goals. So how flexible can we be, and how reproducible is that data? Flexibility, maximize the utility of each technology. The fact that with the raindrop that we can validate our next-gen sequencing panels is a huge advantage. The efficiency, workflow implementation. So when you're dealing with lots of samples, it needs to be simple. How scalable is the technology? And then also cost is a driving factor for enabling global implementation. So our study design for this evaluation was pretty simple. We looked at colorectal tumor biopsies. We were very interested in different kinds of tissue. So we had a number of quad sets of samples, tumor versus normal adjacent, and we also looked at fresh frozen versus FFPE. Every day we get asked questions, say, hey, I have some fresh frozen samples, some FFPE samples. Of course, we can combine them, right? And we can compare the analysis across them. I don't think that question will ever stop being asked. Minimum sample input was used for all technologies. For Thunderbolts, we used 10 nanograms or less. For the Ion AmpliSeq, we used 20 nanograms or less. And then we were required to validate by digital PCR for some of the targets that we saw. And this is just an excerpt, a piece of data from TCGA back in 2013. So instead of looking at all of the things that we found, let's just focus on some of the mutations in KRAS and P53, which are high incidence in these colorectal cancer samples. But before I show you the data, one of the really important things to underscore are some of the workflow considerations. Again, if you're not dealing with large sample numbers now, you very well may be in the future. And this becomes a really large issue for us. So the technology is highly scalable, about 4.5 hours of hands-on time, a total of about 12 hours to get to your sequence-ready library. By comparison for AmpliSeq, there's a considerable amount more time. And this kind of adds up for the numbers of samples that you run. But one of the things that was really striking to us was the time to technology implementation. Literally in 2 days, all of the Thunderbolts enrichment was done and on the sequencer. It took us about 3 months starting with AmpliSeq some time ago to get to that same point. And even though these technologies continue to evolve, really we find it's the simplest ideas that work best. I don't think there's anything that we could do ultimately to improve on that. So this is some of the performance data, looking at the Thunderbolt's cancer panel results versus the AmpliSeq in those same samples. And this is really consistent with what Rupam had shown. This is obviously our data from our samples. We did see an increased number of reads per target, which was nice. And then all the way up through 100x, at least for the Thunderbolts, we saw 100%. Up to 500x, we saw very good results. We're obviously very excited with the latest revision of the panel to be able to achieve even a greater significance for performance, and our uniformity was outstanding. And ion AmpliSeq works just fine. It's a different workflow. It's a little bit of a different approach. However, the performance, albeit what we had set was originally the gold standard now, is just not performing as well as some new technologies that exist. What you can see here is a difference at 500x, but more importantly, the uniformity. And you can see whenever you start to see differences, it's usually in FFPE. And that's the predominance of the samples that we're running for this technology or have been. And if you look at the tumor versus normal, the uniformity drops off. So if we look at some specific mutations that we were interested in, here's one of the KRAS mutations. And the other thing that is interesting is the ability to use digital PCR again on the same instrument to be able to see how tight those variant frequencies are. So again, the AmpliSeq performed very nicely. Here's the Thunderbolts, which performed also very nicely. And if we look at the variation frequency by digital PCR, it lines up. But here you can start to see some differences. So here's a sample that also had a variant frequency of about 38. This was very close. The variant frequency for AmpliSeq is not as tightly aligned. And then here is for a mutation that we found in the tissue samples from those same colorectal samples that we tested. And you can see here a tight correlation. And then there is one sample here that we detected with AmpliSeq that we didn't detect with Thunderbolts. So as we look at this, we had to ask ourselves why. And it turns out for this sample, because we required so much more DNA for the AmpliSeq panel, we actually went back to the block and had to take additional slices. And as we all know, if we can't use the same slice for analysis through and through to validation, we potentially run the risk of getting a different result depending on where we are in the tumor or in the biopsy, and that leads a lot of variability into our assessment. And this is something that we're always cautious of, but is an example of, if we can get more out of the use of each individual sample and we're able to capture everything with the first piece, that we don't run the risk of doing that in the future. And then here's some data looking at some P53 variants. So again, you can see the tight correlation between what we identified in variant frequency on the Thunderbolts and what we were able to validate on the Raindrop, and just a slightly different profile when compared to the AmpliSeq variant frequency. So we were very happy with these results, and for the first time, we were really able to compare these variant frequencies on a really more exacting level by using digital PCR as the validation in that same technology. So looking at our DNA QC analytical workflow, there are a lot of labs that do things by hand. This is more along the lines of the workflow in our lab where it's automated from DNA extraction all the way through to actually assessing sample quality using SNP genotyping. And like I had mentioned, there are certain limitations associated with the QC that you do on a sample that may be used for an analysis that has a higher resolution. And this is what a DNA workflow might look like under construction, and this is actually what it is. Where now we actually use the same chemistries for FFPE as well as whole blood and fresh tissue that are mostly bead-based. We have the same technology infrastructure. We're refining the automation associated with how we do it, but this will become an integral part of our process. So not only can we use this technology as a part of our quality control assessment to make sure that those samples are unique, that they're not contaminated, and that they will hold up for sequence-based analysis, but we can also do target enrichment and generate key pieces of data that now will be appended to those samples for distribution with them. And this is something that at the right cost, at the right workflow, can be done on every sample. So we know that there's a need defined for biobanking. That's why we have 15 to 20 million samples under management. But technology integration is critical for molecular profiling on an enterprise level. And this kind of analysis is going to be pushed at the level of the biobank and the program so that people will be able to get this data to facilitate and move on with a deeper level of research on every sample. Specificity and sensitivity is mission critical. Being able to do the analysis and validation on a single instrument is a bonus, so this integration is really important. The efficiency and cost is operationally essential, and that will be really critical in helping us do this on an enterprise level. And what we found is that the Raindrop technology enables us to integrate sample QC, targeted molecular profiling, and validation in a single platform, which again, for a lab that deals with this kind of a sample flow is really critical for success. So I just want to thank some of our collaborators from CINJ, Bob DePaula, Sridhar Ganesan, and Lorna Rodriguez, who collectively run the Cancer Institute as well as the personalized medicine and translational initiatives. Jay, Mike, and Wasi from the RUCDR. And with that, I'd be happy to answer any questions. Thank you.
Thank you, Andy. I think we may hold questions till the end, and then we'll bring each of our speakers back up. So without further ado, I'd like to introduce Dr. Andy Rhim from the University of Michigan. Andy runs the Pancreatic Cancer Center at Michigan and previously had been at Penn. Focus obviously in pancreatic cyst lesions. So with that, Andy, over to you.
Thanks, Rhim. I'd like to thank Rhim and the folks at RainDance for the opportunity to share with you some exciting data, some of our very recent personal experience using Thunderbolts to assay ultra-low input, to perform ultra-low input target analysis for pancreatic cyst lesions. So our laboratory is interested in understanding the biology of early pancreas cancer with the hopes of using this knowledge to come up with better ways of achieving earlier diagnosis of pancreas cancer and hopefully impact treatment. Now, there are two different precursors to pancreas cancer. The first is pancreatic intraepithelial neoplasias or PANN lesions. These can lead to ductal adenocarcinoma, but they're currently undetectable using current clinical assays such as CT scans or MRIs. So here the problem is detecting the PANN lesions in the first place. But the opposite is true for the second type of major precursor to pancreas cancer, which are pancreatic cystic lesions. And here we're focusing on intraductal papillary mucinous neoplasias, or IPMN lesions. These lesions can lead to cyst adenocarcinomas or ductal adenocarcinomas. But with these lesions, because of the fluid-filled components, and because they usually present a little bit larger, 0.5 centimeters to 3 centimeters, they're readily detectable on CT scans and MRIs. The problem here is that IPMN lesions are being diagnosed at a skyrocketing rate. With every person that gets a CT scan or MRI, we're finding incidental diagnoses of IPMN lesions, such that we now diagnose IPMNs in about 1 in 1,000 Americans over the age of 60. And this is a clinical conundrum because only a small portion of these lesions will go on to develop pancreas cancer. But if we can catch these lesions early enough, we can prevent cancer from forming in the first place by performing a pancreas resection in the form of a distal pancreatectomy or a Whipple procedure. But these procedures are very complex surgeries associated with complications and morbidity. Many patients after this surgery will develop brittle diabetes or malabsorptive disorders. Now, clinical guidelines do exist that let us know which patients we should be referring for pancreas resection. These criteria are called Sendai criteria, and they're basically a consensus opinion of a panel of experts. On balance, there's very little clinical data for its use, but it's the best thing that we have so far. These criteria basically are based on the radiographic appearance of these lesions on CT or MRI and cytopathologic analysis of the cyst fluid. But these criteria are fraught with problems. First, we know that these criteria have very poor sensitivity. Of all lesions that are Sendai criteria negative or thought to be benign, we know that about 10% will go on to develop cancer. But even more worrisome is the fact that these criteria have very poor specificity. So of all IPMN lesions that are resected based on Sendai criteria, only 60% will have dysplasia or invasive carcinoma on the resected cyst. That is to say, about 4 out of 10 patients may be getting their pancreases cut open for no good reason. So we need to develop better ways to predict which IPMN lesions will go on to develop into cancer. And we've been encouraged by recent data from the Vogelstein and Kinsler groups, in which they've been able to show that the presence of a mutation in KRAS and GNAS are highly associated with advanced pathology. And the question that we've been asking in the laboratory is, can we use this information now to predict which IPMN lesions will have invasive or dysplastic pathology in real time prior to the decision of surgery? But there are many obstacles in the genetic analysis of cyst fluid. First, the sample is extremely limited. A 1 centimeter lesion will typically yield about 1 milliliter of fluid. And most of this fluid is already spoken for. So based on send-out criteria, we have to send out all specimens for 2 tests. A cytopathologic analysis, which comprises about 0.5 to 1 milliliters of the fluid, and another 0.5 to 1 milliliters for tumor marker analysis. So it leaves very little in the way of extra sample for us to be able to do clinical trials to see whether or not some of this technology can actually work in the clinic. Moreover, work from a collaborator at University of Michigan, Michelle Anderson, has shown that cyst fluid from the pancreas contains a number of PCR inhibitors that prevent optimal PCR reactions from moving forward. So these were the reasons why we turned to the Thunderbolts, and I won't go through all the reasons why we decided to turn to Thunderbolts, except to say that sample inputs was the main reason why we wanted to use this technology. So we performed a pilot genetic analysis of cyst fluid. This is data driven mostly by my postdoc, Angelina Landana, who's in the audience. And here the hypothesis is very simple. Can we find a genetic signature in less than 10 microliters of cyst fluid that can distinguish between benign and advanced pathology in resected cyst lesions? Can we beat the current clinical criteria today? So we analyzed 5 cyst fluid samples from patients who eventually underwent pancreatectomy. We loaded 10 to 50 nanograms for analysis, and we confirmed mutations using Droplet digital PCR using TaqMan chemistry, as was discussed before. And then we compared the genetic signatures to resulting pathology that was found in the resected pancreas. So here are the standard quality data. These are the 5 samples, and sample 2 has as little as 8 nanograms of DNA. And no matter if we look at the 50 nanogram input or the 8 nanogram input, we get very similar data. We get very uniform coverage of the sample, and we get very deep coverage at that, with almost all samples achieving 200x coverage with the Thunderbolts-enabled sequencing. What about mutation calling? So here we see the 5 individual samples organized in terms of pathology. So if you look at 4 and 5, these are the cyst specimens that had normal or benign pathology. We didn't find any mutations. Well, if we focus on samples 1 through 3, which had invasive carcinoma or dysplasia, now we begin to see that all these samples have detectable KRAS and GNAS mutations. Moreover, we find additional mutations that have been annotated previously but of low penetrance in the population in the sample with invasive carcinoma. We were able to validate these mutations using Droplet digital PCR with TaqMan chemistry, and shown here are raw data validating this. So here we're looking at sample 1 with the invasive carcinoma, and we find that 26% of all the KRAS DNA contain G12D mutations, whereas 0% of the KRAS DNA in sample 4 with benign pathology contained this mutation. So we were able to confirm KRAS mutations in all 3 samples in which we were able to find it in Thunderbolts-enabled sequencing. But what was really striking was the fact that we saw high concordance in the allelic frequency of the KRAS mutations, whether we used MiSeq sequencing data or the TaqMan assay. And finally, we compared our data to a currently widely used send-out test at a clinical laboratory that specializes in the detection of KRAS mutations in pancreas cyst fluid. And what we found was that this send-out laboratory was only able to find KRAS mutations in 1 of the 3 samples in which we were able to find KRAS mutations using 2 complementary technologies. So in terms of future directions, we're really excited with this product because we're getting quality data off of very difficult patient specimens. But this Thunderbolts panel, while great, is missing some of the mutations that we would like to be asking for. So we're working with RainDance in customizing a Thunderbolts panel to make it a pancreas-specific panel at that. And once we have this panel, we have buy-in to perform a clinical trial to test the clinical performance of Thunderbolts-enabled sequencing, as well as a targeted mutation assay for KRAS and GNAS mutations, and compare them to Sendai criteria. So I've shown you data to suggest that Thunderbolts-enabled targeted sequencing can be done with as few as 8 nanograms input DNA from a very difficult specimen, pancreatic cyst fluid. And we're able to show that KRAS and GNAS mutations do indeed correlate with the presence of worrisome pathology in cystic lesions, now allowing us to launch into a clinical trial. So with that, I thank you for your attention, and I think we're taking questions later on or right now? Later on. Okay, thank you.
Thank you very much, Andy. We're going to hold questions till the end. I'd like to invite now Fraser Simons from MD Anderson. Fraser heads the breast pathology and cytopathology group at MD Anderson, and here he is.
So I'm Fraser Simons. I'm a pathologist. I direct research operations, and I wear a new hat as well, in that within the clinical trial cooperative group structure, I lead the translational research program for the Alliance. So the interest in the future application of technologies such as this to clinical trial biospecimens with low amounts of archival material is of great interest to the clinical audience, some of whom I'm sure are represented here. What I'm going to show you is a very recent pilot study, which we were invited. Our group works in RNA gene expression, principally working on development of predictors for outcomes in breast cancer. And the model that we chose for our pilot analysis is just to see the feasibility from a research perspective of this technology to archival DNA, which has been preserved after RNA extraction for our studies, which we've historically done and currently do with gene expression arrays. Secondary to that is can we think ahead to a future where we are looking at more of a clinical trial tool where we can get into better precision and a higher signal-to-noise ratio for our signatures. And thirdly, is there potential to be able to translate this to the formalin-fixed paraffin-embedded environment, again in the clinical trial archival setting and also in the standard routine pathology diagnostic setting. So the model we're going to talk about is triple-negative breast cancer, just very briefly. This is the type of breast cancer that has neither of the hormone receptors and lacks HER2 overexpression. Consequently, it is the least differentiated form of breast cancer and the most clinically aggressive form at a biological level. Its natural history is somewhat more aggressive than the other forms of breast cancer, characterized by early risk of relapse. However, just to add a little complexity and interest to this story, it's also potentially the most curable form of breast cancer by chemotherapy. And what I'm showing here is a system that we developed that's freely available and widely used now called residual cancer burden. And this is a way for pathologists to quantify how much cancer is left after the chemotherapy is being given and the patient then moves on to have her surgery. In pathologic complete response, PCR, different context, same acronym, is the black curve. This is complete remission in the lymph nodes and in the breast. Residual cancer burden 1 in purple is a minimal amount of residual disease, and moderate is 2, and that's red, and extensive residual cancer burden is the blue curve. And you can appreciate that the black and purple curves constitute half of the patients. So essentially, for all intents and purposes, nearly half of the patients with triple-negative breast cancer are actually cured by standard chemotherapy, and for the other half, the red and blue curves, you can see there's significant residual risk despite all of the best current available treatment. And so this sets up a really important scenario for predictive diagnostics. And we've taken this on from the multi-gene array-based platform, training on endpoints such as this, and trying to piece together the various components of prediction and resistance that could lead to an understanding of residual risk after the current planned therapy, and potentially in the future leads to decision-making about whether or not to participate in a clinical trial from the outset of new therapies, and potentially even give you some insights as to what sorts of therapies you might want to take on if you are considered to be still at residual risk. These decisions can be made in a predictive medicine scenario from the beginning before treatment begins. So the three levels we have used in this model were a proof of concept study, where a predicted sensitivity index to endocrine therapy, which is really not relevant to triple negative breast cancer because they're pretty much all predicted to be insensitive since they lack the receptors. But then at the levels of chemotherapy, training on response and training on early relapse to develop empirically multi-gene predictors of expression related to resistance. And then at the third level, training on levels of exquisite sensitivity to chemotherapy where it's either completely removed by the treatment or there's only minimal residual disease. And when we published on an independent validation cohort, on the right are the triple negative breast cancers. 80% of them achieved a pathologic complete response, and their survival curves are shown on the right, wherein those who were predicted to be treatment sensitive by the multi-gene index, that's the red curve, had a markedly better survival than those who were predicted to be treatment insensitive. And notably, the black curve, the predicted treatment insensitive, had a very high rate of early relapse after current chemotherapy. And so therefore, this sets up the potential for clinical utility. So with that in mind, we take some of these samples where we've been working on RNA over the years, patient by patient, and a postdoctoral scientist in my lab, Rosanna Lau, who's in the audience here, found some of these samples, extracted the DNA from the flow-through after the RNA had been extracted years ago, and then we kept the flow-through that had not bound to the silica membrane and just kept it in the -80 for years and years and years. And she dug around and found 8 triple negative breast cancers that had either done very well or very poorly, and then extracted secondarily the DNA. We sent 500 nanograms quite some time ago to our friends and colleagues at Yale University, Lajos Pustai and his group. And then just a few weeks ago, actually less than a month ago, we sent 50 nanograms to be evaluated with the Thunderbolts cancer panel from the same 8 tumors. So we haven't had time to make the comparisons yet, but I just wanted to present the breakdown data of what we found in the first 8 samples of this old, poorly preserved, shall we say, in terms of this is not the way you would want to keep your DNA, samples from our old studies. And what we found here at the listed 8 samples here, as you can see, the lowest is 2,000 reads per target, but most of them are in the 5,000 to 7,000 reads per target. And at 30x, all of them hit 30x, and all of the tumors had greater than 98% at 100x, which is pretty good. And you can see that with one exception, the uniformity is excellent from these samples. In terms of the mutations, as you will have gathered already, we don't have the germline comparison here, so we're really just showing the observations from cosmic annotation for some of the putative mutations that were observed in P53, PIK3CA, interestingly for KIT as well. And then this is just a visual to illustrate that indeed, of course, we picked up SNPs, well annotated here in either homozygous or heterozygous in most of the patients. and a couple of well-annotated mutations, and 2 that, at least at first pass looking at the data, were not annotated in the COSMIC database. So with this in mind, our future applications, we think, are we have the potential with our clinical trial samples to go back and perform efficiently and with a good degree of penetration DNA mutation testing. We think that's a promising use of residual samples. And we're very interested in the possible ways in which the RNA-seq might meet our needs in terms of being able to get at a high number of reads per target. Can we get our gene signatures to a place where they have tremendous precision and reproducibility above and beyond what we can currently achieve? We think this is a very promising technology in order to achieve this. So with that, I'd just like to close and thank you for your attention.